Good morning, student. In previous lecture we have studied duplication, that is one of the type of structural chromosomal aberration. Now we'll see next type of structural chromosomal aberration, that is inversion. Here when a segment of a chromosome is oriented in reverse direction, then such a segment is said to be inverted and the phenomenon is called as inversion. Okay, the gene sequence in an inverted segment is exactly opposite of that in its normal homologous segment. Okay, so here that particular gene sequence, okay, orient and reverse direction. There is no duplication. In duplication there repetition of particular sequence of gene, um, and sometime it get reverse and direction, that is called as reverse tandem duplication. But here only this particular sequence of gene get oriented in reverse direction, that is called as inversion. That get inverted, say particular segment of chromosome get inverted, that is called as inversion. The gene sequence in an inverted segment is exactly opposite of that in its normal homologous segment. Okay, in this case the fragment attached to the original chromosome but in reverse orientation. Then inversion is the reversal of linear order of chromosome segment and its gene sequence. Then chromosomal inversion may be paracentric or pericentric. And before going to see types of this particular inversion, okay, we'll see this diagram. This is original chromosome and mutated chromosome. Okay, this original chromosome has particular sequence of color green, then brown, then blue, and uh, this particular color, that is uh, lavender color. Okay, then this orientation of gene has first green, then coming of blue, and then brown. Okay, this is orientation of particular segment of gene and reverse direction. Okay, then origin of, before going to see type of this particular inversion, we'll see origin of inversion. This inversion occurs when parts of chromosome become detached and turn through 180-degree and are reinserted in such a way that the genes are in reverse order. Okay, some inversion presumably result from entanglements of the threads during the meiotic prophase and from the chromosome breaks that occur at that time. For example, suppose a certain segment broken in two places, the two breaks may be in close proximity because of chance loop in the chromosome. When they rejoin, the wrong ends may become connected due to turning of the detached part through 180-degree. The part within the loop thus get turned around the invert or inverted. Okay, it is known whether all inversions occur in this way, but this is mm, plaus- uh, um, plausible explanation for many chromosome inversion. Okay, here we can see origin of inversion. Okay, this um, then next is type of inversion. Okay, there are two types of inversion: paracentric inversion and pericentric inversion. In paracentric inversion, both breaks occur in one arm. Centromere is not involved. Okay, here inversion occur in both arm only, or inversion occur both breaks in one arm and there is no involvement of centromere. Okay, in pericentric inversion, breaks occur on both arms, centromere as involved. Okay, so this is paracentric and pericentric inversion. You see diagram of this both inversion, paracentric inversion. Okay, in paracentric inversion you can see here that breaks on chromosome, chromatids of chromosome takes place on one arm only, and this particular break part get orient and or reinserted and reverse direction on this particular region. Then pericentric inversion. This breaks occurs on both arms that involvement of centromere, okay, and this get reinserted and reverse direction, okay. This is paracentric and pericentric inversion, okay. Then we have to remember that in paracentric inversion there break occur on one arm only, there is no involvement of this particular centromere, okay. But in pericentric inversion breaks occur on both arms of chromosome and their involvement of uh, centromere, okay. Then types of that effects of inversion, or before going to see effect of inversion, we'll see cytology of inversion. Inversion may survive the meiotic process and segregate into viable gamete, or may form non-viable gametes. During crossing over, if the inversion has been heterozygous, the homologous in uninverted chromosome from a loop like the inverted chromosome, but in a reverse direction the corresponding part come together, okay. Then this crossing over in paracentric inversion will result in the formation of dicentric chromosome, which at anaphase will form chromosome bridge, and acentric fragments which at anaphase will form laggard, and original chromosome and inverted chromosome. Then crossing over in pericentric inversion result in the formation of chromatid with both duplication and deficiency, okay. Then we'll see effects of inversion. Genetic effects of inversion. First is sterility, then crossing over suppression. Then this inversion helps to retain the original gene combination by reducing the crossing over frequency. Inversion enhances the position effects and thereby produce phenotypic changes. Inversion causes chromosome polymorphism within a population, leading to karyotype evolution. Inversion produces balanced gene complexes through reduction of crossing over, which in turn confers great biological fitness and adaptability and increases the evolutionary potentiality. Then this inversion showing or its that genetic effects of inversion, that is sterility. The heterozygote for paracentric inversion are highly sterile and produce only parent like progeny. The dicentric bridges breaks anywhere, and the resulting fragments containing duplications or deletion, the acentric fragment is lost, and that is called as laggard. Therefore, half the meiotic products are not functional. Of the remaining one-fourth of them are functional with normal chromosome, and one-fourth are functional with an inverted chromosome. Considering pericentric inversion, half the meiotic products will be non-functional and inviable uh, due to presence of duplication and deletion in them. The other half of the gametes are functional, one fourth of them have normal chromosome order, and one fourth have the inverted arrangement. Okay. Then another genetic effect of inversion, that is crossover suppressor. The inversions heterozygote often have mechanical pairing problem in the area of inversion. This also reduces crossing over and recombinant frequency in the vicinity. An inversion associated with well adapted linkage group may increase the probability transmitting such a group of gene intact to future generation. The crossing over suppressors can also provide a genetic basis for reproductive isolation between populations, thus playing a role in species formation. Chromosome with inversion have as the chromosome that can be placed opposite to homologous chromosome carrying certain genes that are homozygous. Lethal. crossing over is suppressed in such chromosome and it possible to maintain a gene in the heterozygous state that would have caused death in the homozygous condition. Then laboratory stock carrying several mutant are kept intact without crossing over as balanced heterozygote. Okay. Then this inversion playing important role in evolution. Pericentric inversion appear to have played a significant role in the evolution of humans and great apes relative. The most common pericentric inversion as seen in chimpanzees and human for chromosome number 4, 5, 9, 12, 15 and 1. A paracentric inversion can be seen in chromosome 7 of chimpanzee and gorilla. Both pericentric and paracentric inversion have occurred in chromosome 16 of human and gorilla and chromosome 3 and 17 of human and uh, orangutan. Then this is what effect of inversion. Then next is translocation. Okay. In this way we have seen that another type of um, structural chromosomal aberration. Now we'll see next type, that is chromosomal translocation. The translocation is the shifting or transfer of part of chromosome or set of gene to a non-homologous chromosome is called translocation. There is neither any loss or addition of genes during translocation, but there is only rearrangement of particular chromosome. There are different types of translocation: simple translocation, shift translocation, and reciprocal translocation. Okay. So and next lecture we'll see this particular type of structural chromosomal aberration, that is translocation. And up to this lecture we have seen three types of structural chromosomal aberration: deletion, duplication and inversion. Thank you.